Well, thank you very much I'm always glad to be here at the Institute. So I'm going to talk about evolution in real time. So in the previous talk, we were saying what are the problems of getting just information that is just information that we have right now in the human population and try to go back in history. So I'm going to talk about particular models that they work with real-time evolution, where we can sequence how the genomes change in real time. I'm going to show several examples and how we can extract patterns from this evolution, how looking at the genomes and trying to understand how these genomes fit in a big space. Okay? This is the work of mainly one of my students, Joe Chan, that is an MD PhD and probably the only MD in the US that knows about persistent homology. And then Gunnar Carlson, that is in Stanford, and myself, that is some of the ideas. So I'm going to start with the only figure on the origin of the species in 1859 by Charles Darwin. And he was proposing in this figure how species are created. And the idea is that you have some individuals here, produce some offsprings. Natural selection is killing most of the offspring. There's only a few of them that survive. These produce some children. These children, most of them die. Some of them survive. And at the end of this process, what you have is a hierarchical structure where you have individuals, different species that are generated related by common ancestors. Okay? So this is kind of the tree paradigm. This is how we think about the species and how we understand life in the big picture, the tree of life. So everything was kind of not very informative till the 70s when we started to get good information about sequences of genomes, sequences of proteins, and then we can start comparing these things. And then we can try to basically populate the tree with different species and different organisms and different taxa and trying to see how these things worked. In 1977, using ribosomal RNA, was starting to put together all these cells, all these different forms and species, and he realized that there was something in the middle between the eukaryotes and bacteria and kind of a new domain of life. And since then, this tree has been growing more and more. Okay? So as you probably know, now and from our previous talk, now we have an explosion of genomic data. I mean, genomic data is everywhere. We are getting more and more. It's cheaper. But the problem is, what do we do with this data and how we interpret this data in a meaningful way? How do we look at these patterns of data in a big picture? So there are three kind of philosophical big concepts that are derived from a tree paradigm. The first one, that even if all the organisms can be classified in different taxa, there are some very specific taxa that are called species, where individuals from the same species they can mix, but not individuals within different species. So you cannot have something like, if you try to mate with a very different species, you probably will not have offsprings. Okay? So the idea is that you can have some well-defined taxa where the individuals can mix and produce progeny. And then the tree is reflecting, the end of the trees, the leaves, they are reflecting species. Okay? The other thing that is important also, that is there are some essential genes or some genes that if you just take the phylogeny of gene 1, you get the tree. If you get the phylogeny of gene 2, you have a different ones. You have a different tree, but it's going to be almost the same. So it's going to be kind of a tree plus minus statistical fluctuations. Okay? And then the tree of life that you get, it doesn't matter very much what you take, because at the end, everything is kind of very similar. Okay? The other thing that is also can be deduced from a tree structure is that if all the organisms, they have a common structure, and then this structure is like a tree, you can start merging 2 by 2, and at some point you have something that is the common ancestor to everyone. Okay? So this is the last universal common ancestor, and then there's some speculations. Even Darwin, on the origin of the species, he was mentioning these things. I mean, that if there is something common between all different species, and we go back and there is a hierarchical structure, then there is a common ancestor to all of us. Okay? So we already knew, even Darwin, he was mentioning in a chapter about cases of hybrids, for example, in eukaryotes. So here I'm going to present, let me tell you what I'm going to present, the 3 domains of life, eukaryotes, bacteria, and archaea, and something that is not present in the tree of life, but is the most common form of organism that is on Earth, that are viruses. Okay? It's estimated that there are more viruses than any other species. There are like 10 to the 31 viruses on Earth, but they are not fitting in this tree of life. They are kind of individuals that are kind of out. Even the classification of viruses per se is very complex and is unresolved. So in eukaryotes, we already knew that there are some examples that they are of hybrids. I mean, two different species, that they are different, but they can produce a progeny that can reproduce. In plants, that happen very often. And now with genomics of plants, we are getting many different examples of species of plants that are coming from merging of two different species. Mm-hmm. So there are some very interesting examples that they are done in the lab, and they work. I'm not going to go to the historical description. In bacteria, this is something that is already very known for a long time ago, that there is horizontal gene transfer. There are many mechanisms of taking a gene from a bacterium and giving to another bacterium. And this is a very active field of research. I mean, from the practical point of view, for example, in the hospitals, I mean, many people are trying to look for these kind of events, trying to resistance to different antibiotics and different things. So then we know that in the past, there was big merges. So two different species, very, very different species, like the ancestor of the eukaryotes and a bacterium merge, and they fuse the genome. So there was a huge merging of genomes. And this is how we are here. I mean, eukaryotes, we have the mitochondria, that is a small part there, and it was the result of a big fusion of two different events. Okay, archaea, that was the new domain of life that was identified by Woos in the 70s. Now there are many papers, and now if you go to many of the journals in sequencing, in genomics, and the archaea, they are being sequenced, and these archaea, usually, many of them, they live in very extreme environments, and they exchange a lot of genomic information with bacteria, that they are very different. For example, there are many genes that are going from one to another, and these things. So then, let's go to my favorite topic, that they are viruses. Okay. Oh, sorry, before.
So this exchange, how is this exchange done? I mean, is it through viruses, or how is
well, in these things, I mean, there are many, many different examples. In these cases, I don't think it's through viruses, because there are some big regions that happen there. So another example that was a very interesting one last year. So there are some parts of the human genome, some elements that happened, for example, in where is that? In the gonorrhea, in the clap, in the bacterium of the clap. Probably is known for some of you. And then, so 10% of these guys, they have a human gene that is kind of a retroelement. So these are things that are happening between the three domains of life. The whole structure is getting very messy. It's not looking like a tree. But then something that Arnie, for example, and colleagues discovered here is that in our genome, if we just take a random piece of our genome, the probability that this is a gene that is coding is only 1%. It's only very small. There are tons of things that are repeated elements. There are tons of viruses that are, some of them dead, some of those semi-alive. And they were doing archaeology in our genome, and they discovered genes that were from viruses that were integrated like, I don't know, 30 million years ago or something like that. So it's a fascinating thing because we can do archaeology in our genomes. I mean, looking at pieces of genomic material that are coming from other places and are integrated in our genome. So phages, there was going to your questions. There are tons of exchanges between bacteria and different bacteria through phages. There's a very interesting story with the mitochondria and one of the polymerases that nobody exactly know where it's coming from. So if it's not from a bacterium, and it looks like more to some of the phages. And a very clear important case, it was the Zika toxin in the E. coli in the German outbreak a couple of years ago, where there was a piece of the genome that was related to, it was a toxin, and it was probably coming through a phage. So basically, we are getting now with genomics, we are getting tons of examples of a genomic exchange, and the structure gets very, very complex. So this is a tree that a very nice kind of perspective paper by Tuliten in 1999, where he was saying in the beginning of the 90s, this is what probably most of us studied in every book of biology, is that evolution looks kind of a common ancestor here. Then it's like a tree with the three domains of life. And then there were kind of exceptions to the tree, that they are kind of these branching events where you can have big mixing between different bacteria going like the chloroplasts or the mitochondria at some point. So this is the thing. But at the end of the 90s, I mean, and now I think it's even more, the structure of the tree looks more like something like that. I mean, like there are tons of exchange and these kind of things. So the question here is that if we want to understand evolution in a way that is not a tree, I mean, what can we do? I mean, can we start doing many of the things? I mean, we are doing many things. Taking a tree, we're saying, and trying to put together trees and trying to see how these trees, they are working. And can we find representations that can capture and represent important properties of evolutionary processes? And from the practical point of view, I work with genomes every day, so I have tons of genomes, and then I don't care if they are coming from different species or not a species. I don't want to go into the debate of what is a species or not. I want to find relationship between the genomes, independently of they are coming from the same species or not, and all these things, okay? So I just give you an introduction of what is the problem, something that, probably many of you are aware. I'm going to go to some particular idea that we have of how do we look at this problem through persistent homology. I mean, I know that there were some people here last year working on persistent homology, and probably you know about these things. Then how we can map a problem in genomics to persistent homology, and then how can we read properties of evolution through persistent homology and the other way around, okay? So, and then we are going to my favorite organism, that is flu, influenza. And then I will briefly mention other examples. And after the talk, I mean, we have tons of things. I will be happy to discuss with you about other things. So in persistent homology, this is one of my mistakes. Okay, the idea is that we have some space and we have some points in a metric space, so we have distances. Then we fix a distance, so we have a rule, and we say that 2 points, they are close to each other. Oops. If they're closer to that particular scale. And then we say that 3 points, they are closer to each other if they are all 2 by 2 close, and all these things. So you construct a complex with a particular scale. And from here, you just compute some groups, and you just do your homology. Okay? The idea is that we start with some points, these things. This is your data point in 2 dimensions. Okay? Then from here, you just inflate the points, and then you create balls. And from the balls, you just can create a complex here. And from the complex, you can just compute the number of holes, and the number of triangles, and the number of points that are connected, and all these different things. So, through one example, I'm going to go through one example here. So here, for example, we have this octahedron. So is that they have 6 points here. We just, it's not an octahedron, it's just 6 points. You just take a scale, you start connecting the different things. So if the scale is very small, this point, they are independent. And then you have 6 independent points. There are no loops in 1 dimension. There is nothing in 2 dimensions. Then you start inflating the points. You construct an empty tetrahedron where every point is connected, and there is kind of something in the middle that is kind of a void. And then you continue to increase the scale, and every point is connected. And then what you have is only one big connected component. Yeah. So here is an example on how this is summarized. So let's start with these points in this space. And this is the scale, okay, where we are going to look at the different objects. Here is the 0-dimensional. So this is the number of connected components that are there. This is the number of 1-dimensional loops that happened, the number of 2-dimensional loops. So if we start with this scale, for example, these points, they get connected. The other points, they are at a distance that is bigger than the particular scale. And these are the number of connected components that appear. Okay? So it's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 components. 1, 2, 3, 4, 5, 6, 7, 8. No. 3, 7, 8, 9, 10. 3 and 3, 6, 9. There's kind of. Yeah. Okay, 7 is correct. 3 and 3, 6. 3, 9, 10. I don't know. There is a match in here. Okay, so there is, you match this one. This is the number of connected components. Then you start increasing the value. More points, they are getting connected. So some of these bars, they are going to disappear, corresponding to the components that get connected. And here, the number of components that get connected. And then loops start to appear. So there is a loop that appears here, another loop that appears here, corresponding to these 2 bars. You start increasing. There are 3 loops that appear. 2 of them, they are related to these 2 loops. And then another one. But now every one is a connected component. You continue to increase the whole thing, and every one becomes a connected component, and there are no loops or anything like that. Okay. So now the idea is how we apply that to the problem of the evolution. Okay. So here is an example. This is an example that is very similar to the PCA plot. This one here is very similar to the PCA plot that you were showing. So let's start with a simpler type of evolutionary process that is a clonal process. Okay? So in this model, you can make only copies of yourself, and then you include mutations. Okay? You make a copy and then mutations. So everyone is a clone, and they are related by a tree structure. Okay? Then if you just project in 2 dimensions, what you see is typically you have these blobs. They are related by common ancestry of these guys. And then what you can start doing is just create the persistent complex and try to see what is there. And then what you see is that you have some independent components. These independent components have to merge, and there's a big component. Okay? But you don't generate any loop. Okay? The whole structure is a tree, and there are no loops.
So what is the distance? How have you plotted these points?
This is a PCA plot.
Of what?
Of the distance between any 2 points. So you generate a process. You start with a genome. You start mutating the genome and generating descendants.
Giant distance matrix.
That's right. Well, this is not giant. It's like, I don't know, 100 points or something like that. Okay? So the idea is that you have the process. You start mutating yourself. You generate some children, each of those with different mutations. Each of those generate different children, different mutations, different children, different mutations, and all these things. You take all these distances at the end. So it's very similar to what it was shown before. You just project it on a plane. Okay? And then you try to read what is the persistent complex that is in here. Okay? And then everything can be reproduced by a tree. Okay, because it's a very simple tree that is just looking at who is your parent, and then you start connecting the different individuals to a common original you. Okay, and then everything is a tree that the tree has no loops. Here. Now the second process is very similar, but now you introduce sex here. Okay, and the idea is the following: you just create some children, and then these children they can exchange genomic material where half of the genome or a part of the genome is coming from one of the parents, and the other half is coming from another parent. And then you have another one, half of or a part of the genome is from another one. And this is typical of what you think. Instead of getting just this clump and these blobs, you start getting things in the middle. These things are starting to connect. Okay? And then you can start doing the persistent homology things. You start joining the points if they are closer to some particular scale that is read in this direction. And then you see that there are some components here. The components very quickly connect, and they go to a 0-dimensional one. But then you start getting some interesting structures. So you start getting loops here, all these things. So for example, you can get something like that. The cartoonish picture of that is that you can have children now, and these children, they can generate something here that is different from just a tree structure. Okay. So these are 2 theorems that were proved by, well, the second one it was already proved. The first one it was by my colleagues in Stanford, by Gunnar Carlson. And the idea is very simple. So you have an additive structure. This is a tree where the distance between, so the distance, there are 5 points here. The distance between any 2 points is just the distance through a tree. Okay, so the distance between A and B is 4, 3 plus 1. So you have A and B is 4. The distance between A and C is 3 plus 2 plus 4. Okay, so you go to 9, and you have an additive tree. So you have an additive structure. So you have a distance that is coming through something like that. Okay, something that is coming through a clonal process. If there is something like that, then the whole homology that happens is in 0 dimensions. There are no loops in higher dimensions. Okay. The second thing has to do with the stability. So if you just change a little bit the things and you start moving a little bit and saying that there is small effects like sequencing errors or homoplasies or something like that, then the results are going to be, they are not going to change very much. The size of the largest bottle bar that appeared there is not going to be very large. Okay? So here is one example. This is one of my favorite examples because it's one of the simplest. So the simplest example where you can have different histories. So this is, for example, if we take the mitochondria and the Y chromosome of most of the population here. So you can see that if you take 4 individuals, let's say that we have 4 individuals here in the population, we can have 3 possible different trees that can represent some of the stories. Okay? So if I just take the mitochondria, it could be that the story is that individual A is related to individual B, or it can be that individual A is related to C, or A is related to D. Okay? So there are only 3 different possibilities if there is a tree. Okay? Now, if you just take the Y chromosome, for example, it can be that it's different. So you can have for the mitochondria is this one, and for the Y chromosome is a different one. Okay. So this is kind of a representation. So they have 6 possible parameters that you can have between 4 different species, and the parameters, the trees, they're 5-dimensional. They are 5 different parameters, and you can have these trees all become one topology when the branch in the middle collapse. So this is something like that. I'm not going to go into the details of that. But this is what one example. This is done by Kevin. That is there. So imagine that you have. This is the trivial example. That you have one gene, say mitochondria, for example, has this particular distance matrix. Okay, is following that particular tree. Okay, is tree-like, is fine. When you have a different gene, because recombination happens, they have a different history. Okay, we have 2 different histories. A priori, if I just take these 2 genes together, that this is what most of the time is happening, then you get something that is a mixture of this story and this story. So there is no tree structure that can capture both of them at the same time. Okay? And then you can compute the distance. That is a trivial linear combination of both. Okay? And this is what is doing. This is a nice example. So here is the 4-dimensional. So this is the 0-dimensional, and this is the 1-dimensional homology. These are saying that we have 4 species, and these species, they merge into 2 to 1, and then there is so this to 2, and then it's going to merge into a big one. And here I'm going to show you a video where we are going to do the following exercise. Okay? We are going to start taking a genome that is completely a tree, like that. Okay? And then we are going to mix with some parameter between 0 and 1. 0 means that it's only this topology, and 1 means that it's only that topology. Okay? And then with the mixing, it's going from one to the other. Okay? If you have something 0, then you are completely tree-like. If you have something that is 1, you have tree-like, but a different topology. And in the middle, what you have is a parameter that is interchanging the 2 of them. Okay? Or interchanging, just mixing the 2 of them. Okay? Going back here. So I want you to look at what's happening here when we start mixing. The parameter of mixing is there. Okay. So here, you can see here the mixing happens. The bigger bar happens in 0.5, and then it's going to 0. Okay. So here is just taking, when you have tree-like structure, there is no loops. When there is another tree structure, there is no loop. But if I just take the mitochondria and the Y chromosome together with some combination, you start generating loops. Oh, yes. If I find the pointer here. Here. Okay. Yeah, it's a very interesting example because it's the first. I mean, 4 species is the minimum that you can have different tree topologies in these things. Okay. So the idea is the following. The idea is that in persistent homology, we have data where we have points in a metric space. Then we create complexes. So we start relating 2 different genomes if they are close together. And then we start counting loops and trees and all these things. And then we compute some numbers, like Betty numbers, for example. This is what we are doing. We are very simplistic. When we are working with genomic data, we can think about this process is that we have a very high dimensional space with points that are different organisms or represent the genome of different organisms. Then we have a metric, and we can just create some complexes at different scales. And from there, we can deduce, counting these things, for example, we can compute how many exchanges happen of genomic material at different scales and different things. So I'm going to my favorite viruses, flu. Flu is a great virus. I mean, I was working here when I was at the institute. I was working here with Arnie in flu. We have many papers. It's a great virus for several reasons. One of those is evolving very, very fast. Just to give you an idea, is the first genome that we have is from 1918, from the Spanish flu. As you probably know, it killed many people, like 50 million people in the world. And it was sequenced some years ago, going to some graves. It's a very similar story, but instead of sequencing the person that was there, you sequence the virus that was killing the person. And so they resurrected the virus, and they did many experiments trying to see what was going on. I mean, why it was killing so many people. So So this is the first one that we have in 1918. And then now, I mean, many labs around the world, they are collecting of these things. I think currently there are like 65,000 genomes. So this is of these things. And we have evolution in real time. So we have every year, now we have like every day, we have hundreds of these things coming. And what is great is that the evolutionary rate is so fast. I mean, this virus, every time that it's replicating, it's a few hours, 6 hours or 8 hours, it's introduced errors in these things. These errors accumulate very fast. And then what is happening is that the evolution is so big that you can just, in months, you can track the virus. I mean, one of the things that we are doing is trying to find how the virus moves around the world because the changes there are so fast. It's basically the migration project, but in real time that we have all these viruses. And just to give you an idea, for example, of the numbers, is you just take the virus in 1918 and you just compare with the virus right now. I mean, the virus from the pandemic in 2009, for example, it's a little bit more than 10% difference. So this is similar to a human and a mouse. So it's just taking a picture of a human or a mouse, or the ancestor of both of them, and then trying to see how it's evolving into the different descendants that is there. But this is in real time, so we know this picture in real time. Okay? So this is a fantastic method, a fantastic organism. Then the other thing is that they have another way of evolution. So in addition to the clonal evolution, so this virus, every time that is infecting a new cell, is producing some errors. Infecting a new cell is creating some errors, and these errors, some of them fix, some of those they don't. But they have sex also. Okay? So you can have two different viruses that when co-infect the same cell, they can exchange genomic material. And it happens in a particular way that is called reassortment. So what is reassortment? This is a virus that is segmented. That means that they have 8 different chromosomes. In this case, it's 8 different chromosomes. And the chromosomes, they are here in this cartoon. They have some names referring to the proteins they are coding. When you have 2 different co-infections of the same virus, of different viruses, then you can create something that the PP1 segment, for example, can come from the parent 1, PP2 is from parent 1, PP1 from parent 2, and all these things. So you get a lot of combinatorials of all these things. So this is at the heart of the pandemics. For example, this is what happened in 2009, what happened in 1968, in 1957, and probably in 1918, although we don't know because we don't have any ancestor information. So, so this is fascinating because it has these 2 ways of evolution. So we can test what happens if we have clonal evolution and when we have the reassortment. The third thing that is also great is that we have evolution in real time. So we have these things, how they are evolving. And we have tens of thousands of genomes. So we can just try to look at there. And if we find some interesting pattern, we can go back to the data and we can try to interpret what is these things. I mean, we have good positive controls there. Okay? These are the proteins that I would like you to remember. These proteins that are very important because they are the ones that you hear in the news every day. Hemagglutinin and neuraminidase, H and N. This is, for example, the H7N9 that everyone is scared of the China outbreak, or the H1N1 in 2009. They refer to some proteins that are in the surface, and they are the ones that are recognized by our adaptive immune system. Okay? Typically, a pandemic happens when there is a replacement of these guys here. Okay? In 1968, for example, was a pandemic that was an H3N2 that replaced the previous H2N2 that was going around, and it replaced this protein. So our immune system, our adaptive immune system, haven't seen that thing before. So you don't have any memory of these things. So you are fighting a new thing. Okay? The other things that are kind of important, all the other ones we are not going to mention here, they are these Ps. It's very easy to remember because the P is for polymerase. These are 3 proteins, PP2, PP1, and PA, that form a polymerase complex, and this is the machine that is doing the replication of the virus. This virus is an RNA, and it's a single-stranded antisense. So we are double-stranded DNA organisms, so our genome is 2 strands of DNA. These guys, they don't see DNA at all in their evolution or in the process. I mean, they are not retroviruses. Okay? So to do the replication, they don't use the machinery, the polymerases of the host. They use their own machinery, and this coding by the 3 biggest segments, the 3 biggest chromosomes, that are the polymerases. Okay? So as I was saying, there are 2 very interesting modes of evolution. The first is that just clonal evolution. So you take a virus, start replicating in a cell, producing clones with mutations, and then another one is going to another different cell, clones with mutations, and they accumulate, clones with mutations, and they accumulate. And the evolutionary rate is very high, okay? As I was telling you, it's changing very, very fast. This is great as an evolutionary model because you can just go there and track the virus, how it's going from one place to another, and all these things. And I can tell you tons of anecdotes of students finding interesting things in databases of things that they are not supposed to be there. The other thing is that they have sex. So they can have co-infections of two different viruses, and they can generate this process that is called reassortment. And what you can imagine is that asexual, the drift, is that if you can imagine the evolutionary, the sequence space, all the possible viruses that you can have, when you are doing mutations, you are just moving around a little bit. So you are changing a little bit the genome and all these things. When you have, when there is something that is coming from a sexual reproduction, you are taking two different organisms and created something that is very far away from each of those because it's a combination of both of them. So there are big jumps in the space. So it's a space that is not kind of a continuous kind of a nice space, but they are kind of very complex structure. And what we are doing sequencing, what we are doing is sampling this space of properties. And we would like to understand properties of this space from the sampling, from the genomes that we do. Okay? So this is from the book of Arne, and this is how pandemics they are produced. And this is one example. It was the pandemic in 2009. So you are here, okay? You're a human. And then you have a virus here that is infecting, it's coming every winter, and you get the virus with 8 different chromosomes that they are very well adapted to humans, okay? You get sick, usually you don't die. Many people don't die. In particular, chromosome 4, or segment 4 and 6, they are coding for hemagglutinin and neuraminidase, that they are the proteins that are in the surface. Then there is another virus that is infecting birds, ducks here, okay? You can see that this virus, this is the color here, is very well adapted to birds, and birds usually they don't get sick with these things. But for some reason, these two guys, they came close to another friend here, and they got this guy, and this guy came infected with these two, and it produced something that is a combination of a virus that is 5 parts here, or 6 parts here, that is human, and 2 parts here, they are bird. If this guy goes back to humans, this is a virus that is kind of well adapted to humans, but 2 of the segments, the ones that are coding for the proteins in the surface, the ones that are recognized by our adaptive immune system, they are kind of new. Okay? So this is what happened in the pandemics in the past, these kind of events with different combinations of different segments. Okay? So this is, for example, something that happened in 2009. This is a very interesting story because it's unsolved. There are many question marks here in this picture. This is time here. This is the 1990s, 2000, 2009. That was the pandemic in 2009. As you probably know, it was the first cases that were reported in Mexico. Very quickly in California and Texas, there were several cases. And within a month, it was basically in every continent. So that was the virus here. The color code here represents different segments and the origin of different segments. Okay? So what you can try to do is just trying to infer what is the evolutionary history of this virus. This is important because you try to understand how this virus happened to be and why it came here and where and all these things. And then one of the things that it was clear, it was they have 2 ancestors. One, there was a virus that was circulating in North America, so it was here in the US. And the other, a virus that was circulating in Europe and Asia. But this one, it was in the 90s. So this is one of the parents, and this is the other parent. This parent, it was only 2 segments, the neuraminidase and the matrix, one of the segments. And the other ones, they were coming from these viruses. That these viruses, they were kind of famous at the end of the 90s here. It was called triple reassortments. They were identified in pigs. And pigs, they were found with a viruses that was a combination of viruses that were infecting swine, pigs, humans, and birds. So for some reason, these viruses, they combined. They produce a virus in a pig. And this virus in a pig in North America, for some reason that is still not known, combined with a virus in Eurasia, and it produced something that was kind of the pandemic in 2009. There are many question marks. How these 2 viruses, they met, and how they created these things, where it happens, and how it happens, and we don't know many of these things. Okay? So here, let's say that we just take only one segment. Okay? And then let's try to see what's going on. If you do phylogeny, you get a tree that is very much like a tree like that. If you just do the persistent homology, what you get is that everything is zero-dimensional. There are no loops. Okay? So you can start doing the clustering of the different bars and trying to see who is going with who, and you get something that is kind of very much similar. So this is something recapitulating something that we know, that there is no recombination, homologous recombination in influenza. Okay? So if you just take a segment, the evolution is very much clonal. Mm-hmm. Then what you can do is the following. You can take one segment, hemagglutinin, and everything is zero-dimensional, so there is no loops in the evolution of one segment. You just take another segment, the neuraminidase, and there are no loops. You take the different segments, and there are no loops. When you just take the whole genome together, so it's like taking, this is the X chromosome, sorry, the X chromosome, the mitochondria, and there are no recombination. You take the Y chromosome, no recombination. But now you take everything together, and then you start getting a lot of a structure here of one-dimensional, two-dimensionals, and all these things. So you start getting tons of loops in the whole structure there.
When you put it together, what is the metric? How do you tell that?
It's very easy. You just take the genome of one segment and just concatenate to the other one, and to the other one, to the other one.
Right, but what is the distance?
Oh, the distance is a variation of Hamming type of distance. It's not Hamming. It's just the number of differences with a weight. Yes. Okay. So then you have a lot of structure. So this is a very, very complex space. So the idea is that if you just take only one segment, you have a completely tree-like structure, another segment, another tree-like structure. You put everything together, and you have this very complex thing. What we were very interested is not only just in the loops and the higher dimensional loops that we don't understand many things, is trying to understand exactly what segment is going with what segment. Because there are several things that are very interesting here. When you go to some of the pandemics in 2009, or in 68, or in 57, we realized that not every combination, so you can imagine if you have two parents, that in principle you can have 256 possibilities. You can have chromosome 1 from parent 1 or 2, chromosome 2 from chromosome 1 or 2, so 2 to the 8, these things. The ones that happen in nature, they are not all of those. There are some of those that happen more often than other ones. So we want to understand what are the statistics of how these genes are correlated and segregated together. Okay? So then this is avian influenza. So these are viruses that are circulating in birds. And then one of the things is that there are segments that co-segregate together. They like to go together. And in particular, they are the polymerases. And one protein, the NP, that is also interacting with that. And other proteins, they like to be switched. Some of the segments, they like to go around. Yeah. So here is the different similar plot that what I was showing before. In this case, there are only two segments. Again, this is Kevin. So you have only one segment, hemagglutinin. Okay, that is completely tree-like. Neuraminidase is completely tree-like. And then you start merging the two. In this case, there is a parameter here that is saying what is the fraction of one and what is the fraction of the other one. Okay. And then let me see if I can play. So just look here. Okay. So we start with something that is completely tree-like. We start with hemagglutinin, and then we start combining both. If I can find here, yeah. So you can start seeing that there is a lot of structure that appears when you start combining the two things. There are tons of structure. And then you go back to the other side, 0.8, 0.9, and the whole thing becomes tree-like. So this is another thing that we are working right now. So this is, again, so this is the distribution of this one-dimensional homology. You see that there are tons of bars here. And one of the things that we didn't realize in the first round is that the distribution of bars is not completely random. So we just look at the distributions. Here, this is another way of representing the data. So instead of having a point scale where you have a bar, a particular class that is born, and another one that is dying, you can take these 2 points and represent the birth of the class and the death of the class. Obviously, because the death happened after the birth, then you have something that is above that. Each of these points represents a bar here. Okay? And when you just look here by eye, you can realize that there is something kind of funny because there are tons of things here. There's a structure there. And we just plot the histogram of how many plots there in this direction, in the death direction or in the birth direction, you realize that it's bimodal. So we were looking at that, and this is a very interesting structure. So why is that? And then we realized that it was very interesting because there are these big clusters that are related to the H and N. They are the different subtypes. And it could happen two things. So it can happen that there are mixtures between viruses that are within the same type, and they are very related to each other. Okay? They are kind of the red ones. And there are other ones that happen between different types. So you have, it's like interracial mixing. And these are kind of the blue. So it's very interesting because you can get what is the scale of how this genomic exchange happens. So it can happen at very small scales or very large scales. Okay? So this is, we identified new cases of mixing. This is, for example, a nice one, that it was the first using descendants from the pandemic in H1 in 2009. This is the H1N1 PDM, means pandemic, because there are two H1N1s circulating for a time. And so we look at all these guys, and then we realized that there were some small bars that appear, some mixing between the different things. And that was one of the mixings that it happens to some of the descendants of the viruses here, something that was isolated in 2009 in Athens, in Brussels in 2009, in the Netherlands in 2009, and all these things. So these guys, if you follow, it forms a loop, okay? And is non-structure. And here there are several segments that co-segregate together, and some of the segments that they don't. Okay? So this is another example. This is something that we're very interested in. In the beginning of, it was in mid-March in China, people start getting sick and starting to die from a new kind of flu that was identified at an H7N9. So this is very different from the viruses that we have circulating in humans. In humans, we have H1N1, H3s and 2s, and other types, but it's just these two types, they are the ones that are in humans. This is very different. H7s, this is not circulating in humans. It's very different. The N9 also is not circulating in humans. And there was an old guy in Shanghai. There was another person in Shanghai. And immediately, many other cases start to accumulate from mid-February to May. And that was very interesting because it was not clear what was the way of how this virus was moving around, how people were getting infected with this virus. It was coming clearly from birds because this is an avian virus. It was similar to other viruses found in birds. And it was not clear what new virus was going around. And there were two papers, I think, like 2 days ago in Nature. I don't know if you saw them. They was by the group in Kawaoka in Madison and the other group in Europe, by Fouché, that they were taking these viruses and they were trying to see if this virus is going to become the next pandemic or is not going to become the next pandemic. So they did these experiments that are very controversial, that you take a virus that is not growing, not infecting very well ferrets, that is used as an animal model, and then they adapt it to ferrets, and then try to see if they are able to create something that is airborne transmissible easily. And they try to look for the mutations that happen there. Okay? So this is an interesting one. And when you look at the virus, this is two of the first isolates. This is from the old guy from Shanghai, and the other ones, they are identical. And then when you just look at the persistent homology, you find that it creates this type of polytope in 2 dimensions, so a class in 2 dimensions, where there are 3 parents. Okay? There is one segment that is coming from the H7 and 9 here, another segment from the H7 and 3, and all the internal segments, so the other 6, they are coming from a different thing. Okay. So just talking about other viruses, this is a very different type of virus. This is a retrovirus, HIV. It's very interesting because it's non-segmented, so it doesn't have this property that you have different chromosomes. It's only one genome. It's 10,000 bases, roughly, 9,000 something. And then, but these viruses have recombination. And this is something that is very easy to capture in the persistent homology framework. The thing, if you remember in flu, if you just take hemagglutinin, you have something that is only 0-dimensional. There are no loops, so this kind of a tree structure is there. The same thing if you just take different genes, it's true. But now you take only one gene in HIV, and you realize that there are tons of high-dimensional homology. And this is something that happens very often in HIV, is that you can have recombinations, but the recombinations happen within the segment. Okay? So there are 3 big genes, GAG, POL, and M, and each of those, they have a lot of recombination. So there's a lot of homologous recombination. So you can get a lot of structure. I'm not going to go into the speculations. I mean, we can, if someone is interested in that, there are some very interesting things that are coming from there, and these things. So this is a very interesting structure that we are trying to understand, because there is a big dimensional loop that happens between these very complex recombinants that are coming from some places in Africa. They are super complex recombinants, and there are some speculations by several people saying that these are very ancestral strains. If someone is interested, we can talk later. So you can do the same thing for different viruses, and you can constrain the number of bars that appear in different viruses and different segments, and trying to see how it works. Okay, and just to summarize, is that I was given a talk, an introduction of why I think it's good to try to think about ways of thinking evolution beyond a tree, or a tree with a little bit variations, or several trees, or ensembles of trees, and these things. This is a particular idea that how we apply some persistent homology to these things. I apply to an organism that I know kind of very well. There are tons of data, and we can just try to interpret what is the result of the homology, how the different classes, what is the meaning of that. And there are many other examples that we've been applying to. And again, this is kind of what we are trying to do now, is just trying to detail the dictionary between persistent homology and viral evolution. So if there's some filtration value here, this is a genetic distance that we can just match. The zero-dimensional Betty number at some particular filtration is the number of clusters that happen at this case. The generators of the particular class is a particular representative of the cluster, of the zero-dimensional. The one-dimensional means recombinants of reassortments. And then all things. There are many question marks here that they are not solved, and but they are very interesting, and we have many different ideas in this direction. Again, it's just phylogenetic representation could be very misleading. Someone I was talking, I think was with you, that if you take the data from a mitochondria and then you take from the Y chromosome, you get very different results, and it's very misleading and very confusing when you try to do the same thing. You can imagine that if you just take any particular gene of the genome, you are going to have a very similar story, but perhaps the left part of the gene is going to be a particular story, and the right part of the gene is going to be a different story. So, I think it's good to think about how we understand evolution in something that is a little bit richer framework. And then it's just a different way of understanding these things. The work has been done mostly by Joe Chan. And as I was saying, this is the only MD in the US that knows about persistent homology. And if you have something, probably he will be working on cancer, so he can just compute your Betty numbers in these things. And Gunnar Carlson, that is in Stanford. OK, thank you very much.
Interesting that the dimensionality changes as a function of the different biological processes, one being simple mutation, the other being recombination or reassortment.
Mm-hmm.
You could actually test other biological processes, deletions, inversions,
correct,
duplications.
Yeah. Correct.
Even the fact that the chimpanzee has one more chromosome than we have means that the genome's rearranged in such a way so that the chromosome numbers are non-identical, and yet we talk about relationships between the two species. Since you know those things, those are different biological processes. Question would be, is the dimensionality going to jump to 2 dimensions or 3 dimensions in that kind of process?
Yes, I don't know. It's these things. We are trying to apply that to different processes where the distance is not just an additive distance. The interpretation there is far from clear to me. But there are many interesting processes that are like the ones that you are describing. So, I cannot tell anything about
the
specific.
If it's true that the bar graphs and the birth and death curves, in effect, are giving rise to, at dimensionalities, different biological processes, this is a chance to uncover biological processes we don't know about. I can list the ones I know, but I don't, if you're in the 4th dimension, what does that mean for biological processes?
I don't know. So we are working with viruses because this is the one that I understand very well. And we can generate very high dimensional classes. But I don't know about the other ones. I mean, you're completely right. I mean, it's just completely unknown. Working with human data is kind of a clear direction where, I mean, we don't know exactly how it will apply. Yeah.
So there are no calculations of persistent homology on human data, is that right?
Not that I know. I don't know if...
And a very interesting example might be the percentage of Neanderthal genome in human, because those were thought to be different species.
Yeah,
obviously some subset had sex.
That's right. So I was thinking, for example, one great approach that I think all these big migrations or big mixtures of different things. So in the example with flu, they were clear that there was kind of small mixings that happen intra-subtypes, and there are big mixings that happened between different types. And these are reflected because of the existence of these big bars that happen in the distribution. So I don't know, applying to human data, how it would look like. I don't know. That's something that we have to...
Yeah, the first paper that was cited, that the Lee Durbin paper, precisely is implementing an approach to recombination, but it is not. It's a sequence of trees.
Yes, sequence is tree, yeah.
But not persistent.
No, it's not persistent homology. So yeah, this is one of the things that we have in the agenda in the moment that we have some time and funding. Oh yes, we see some, well, convergence. In viruses, we see a little bit of homoplasy. So there are some mutations that happen in the same thing. So we don't know if it's due to selection or different things. You can see that this is usually reflected, but it's very rare that you have exactly the same type of mutation of many different bases at the same time, unless there is a recombination or something like that. So our interpretation is that small bars can be due to homoplasy or a small recombination, so we don't know. But big bars is only the interpretation is recombination. So there were a few more cases reported, I think, a few days ago, but it's very unclear. It's very interesting. If you go to the Nature, I think it was a few days ago, a couple of days ago, they did experiments on trying to see how, if they are concerned, we should be concerned or not, answer the question if this is going to be transmissible. So all the cases that we know, so this is the official story from WHO and the Chinese government, is that there is only bird-to-human contact. So this is a virus that is transmitted by birds, but it's unclear, and it's completely asymptomatic in birds, so they don't even feel it, and it's going around and all these things. So of the cases that, of these 150 cases that were reported, half of them, they were not in contact with any bird market. This is what is thought that it was the main source. And it was unclear how they got it. But, and then the authorities just went around and trying to see if there were people who were in contact with the person who got infected. And they couldn't find that there was kind of spreading of the whole thing. So what they were saying is that, I mean, there is no clear evidence of human-to-human transmission. In the papers that were reporting these things and doing the experiments a couple of days ago, they showed it was very easy to create aerosol transmission in these viruses. So these viruses, they replicate as, they accumulate errors like crazy. So it's uncertain, what I would say, but I think it's not very unlikely. I mean, it's not very unlikely that it's not going to be a huge jump to become a human-to-human transmission. It could happen. I mean, it's in the neighborhood of the things that could happen. It's not going to happen through a recombination, for example. It could happen directly from direct transmission. I don't know if I was very clear, but I'm trying to. Well, there was one case outside of China. There was the Taiwan. For the Chinese, it was not outside of China. Yeah. So it depends on who do you ask and how international it is. Thank you.